Hello everyone and welcome to today's live broadcast, Intuitive and Easy Programming of Liquid Handling Tasks with epMotion, presented by Dr. Carsten Buhlmann, an International Product Manager at Eppendorf, and Dr. Tim Schommartz, an Application Specialist at Liquid Handling in Eppendorf. I'm Susie Valdez of LabRoots and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots and sponsored by Eppendorf. Eppendorf is a leading life science company that develops and sells instruments, consumables, and services for liquid, sampling, and cell handling in laboratories worldwide. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time during the presentation. Just click on that green Q&A button located at the lower left of your presentation window and type your questions into the box that appear on that screen. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice that you will be viewing the presentation in a slide window. To enlarge that window, just click on the screen icon located on the lower right. If you have trouble seeing or hearing this presentation, click on the support button at the top right of the presentation window or use the Q&A button and let us know you're having a problem. I'd now like to introduce our two presenters for today's webinar. First, Dr. Carsten Buhlmann. Dr. Buhlmann has a scientific background in biochemistry, molecular, and cell biology. He has more than 18 years of experience in life sciences marketing and is working as an international project manager in automated liquid handling at Eppendorf. Our second presenter, Dr. Tim Schommartz. Dr. Schommartz works as an application specialist for automated liquid handling at Eppendorf Headquarters in Hamburg, Germany. He studied molecular life science in Germany and in Singapore, focusing on the influence of anti-inflammatory antibodies on allergies and autoimmune diseases during his master thesis. Tim Schommartz holds a PhD in chemistry, working in the field of virology. He joined Eppendorf in 2016, having a broad background in different workflows ranging from molecular biology, cell and infection biology, to immunology and biochemistry. Welcome, gentlemen. I'll turn it over to you.
Thank you, Susie, for the kind introduction, and welcome everybody for today's webinar. So what I would do is in the webinar we will give a short introduction to automated liquid handling, and as this instrumentation is used in many applications and workflow, we'll shortly explain in what application and workflows the automated liquid handling systems are used in. And then we will explain how easy it is to break it down, the different application and workflow to certain building blocks, and then explain how easy it is to program and handle these building blocks to build up the application and transfer them to automated liquid handling systems. Then we will close in the second part of the talk then with explaining all the various advantages of the automated liquid handling systems, and then close the presentation with a short overview about our epMotion portfolio. So you might know Eppendorf for decades already for handheld liquid handling devices and other lab equipment, but for more than 15 years we offer already also automated liquid handling systems with our epMotion product line. And what these automated liquid handling systems are doing, they are also called pipetting workstations or liquid handlers or pipetting robots. And it's very simple what they do. First of all, they transfer liquids at different volume levels from destination vessels to source. No, the other way around, from source vessels to destination vessels. And this can be from tubes to plates, from plate to plate, or from reservoirs to tubes or plates, and so on. So it's very simple how it sounds. However, in addition, the liquid handling systems can also perform a lot of other capabilities. So They can mix by pipetting or have mixing capability on deck, so that you can do a motor-driven mixing of plates or other labware. And of course, you can also have temperature incubation for cooling and heating of reagents and samples, doing incubations and so on. For a lot of applications, you need to transport the labware plates from one position to temperature incubation position and so on. So we can transport labware on the working deck, and for some applications, we can also do separation by magnetic or vacuum support on deck of the automated liquid handling systems, which is used for nucleic acid purification or other solid phase extraction. You'll see the instruments, the liquid handlers, are quite capable and do more than just pipetting. So with the nice flexibility of the automated liquid handling systems, they can be used for really a wide, broad application area. So on the next slide here, I want to just briefly talk about what are our customers currently doing with our epMotion system. So they use the epMotion system for applications like the next generation sequencing, library preparation, but a lot of customers are also doing PCR setup or nucleic acid extraction for various kits that can be provided from various vendors. And we also have a lot of customers doing cell-based assays, that is more in a plate-to-plate format, or compound screening, ELISA assays, other biochemical assays, SNP assays. So there are really a lot of different applications that can be handled on these automation systems. So there are a lot of application notes available on our website, but you can also screen for epMotion in scientific publications to find really how often this instrumentation is used and how it is used. So before I transfer over to Tim, I would just say or introduce that he will show you now how easy and intuitive it is to program and transfer these applications to an automated liquid handling system. So Tim, please.
Thank you very much, Carsten. So I will take over from here, and I'm sorry. So most of the workflows can be broken down into several parts. So they usually start with a sample, which then goes through preparation or purification processes, which leads to an analysis and which will finally give you a result. And each of those different parts, again, consists of building blocks, which I will come to in a second. And those building blocks, again, consist of several functional steps. So if we, for example, look at a dilution building block, a dilution is a combination of several liquids to reach a defined concentration. And looking at automated liquid handling, we will mainly look at the building blocks of the preparation part, but obviously you can also use an automated liquid handler for downstream processes after you've got your results and continue with subsequent experiments. So let me move on to the building blocks of the workflows. So we divided a workflow into 7 major building blocks, which are reformatting, dilution, cherry or hit picking, normalization, mixing, incubation with or without temperature, and transports, for example, to a vacuum device or to a magnet. I will now walk you through all of those building blocks. Let's start with reformatting. What do we mean if we talk about reformatting? So reformatting simply describes the transport of a liquid from one vessel to another. So those vessels can either be tubes or plates. They can come in different formats, so 1 well to 384 wells. And reformatting is by far one of the most common and also one of the most important building blocks in all of the complex applications you handle every day. So this might sound a little bit abstract, so I've tried to fill this with more life, looking at the everyday example from the lab. So let's say you want to transfer samples from tubes to a plate. In a manual workflow, you would do this with a manual pipette, take out the sample from the tubes, put it in your plates. Or in a more cartoonized depiction, you take out liquids from one vessel and transfer it to another vessel, either in one or in several wells. And how is this transferred into our automation software? There is a very simple command which is called sample transfer. And this sample transfer allows you to transfer samples of different nature. For example, if you do sample replication, plate multiplication, or also sample normalization. So how do we do this in the epBlue software? Let's take a look at the software. This is how the software looks like. And as a first step, you start with populating your work table, which is basically what you would do if you would populate your lab bench with all of the desired labware you want to use for your experiment. So from the drop-down menu in the left, you choose your desired labware, which can be, for example, plates or also tips or any other type of labware. You then choose the specific types of tips, for example, that you want to use for your experiment. So in this case, 50 microliter tips with a filter, and you simply drag and drop it to the work table. You then end up with your tips being placed on the work table. You then step by step populate your work table with all of the desired labware that you need. If you want to change some settings, so you can, for example, change the name of the labware on the work table. Once you are finished with putting and populating labware on the work table and changing the names, you can then simply switch to adding commands by clicking on the Switch to Procedure button. You will then come to another window which looks like this, and there you can drag and drop commands to your procedure list simply by moving them from the menu to the left into your procedure list. We have, in this example, we are using a sample transfer. You then choose a tool you want to use for this process, the transfer type, so either pipetting or MultiDispense, and the volume you want to move. You choose your source and your destination vessel, and you then click on patterns and simply choose the pattern how you want the pipetting process to proceed. So in an enlarged view, you can see here that we transfer the sample from tube number 1 to well A1 of our plate. And we transfer the sample from tube number 7 to well B1 of our plate. So this is a very easy 1:1 sample transfer. But you can also do something like this. So in this case, we do a triplicate. So we transfer the sample from tube 1 to wells A1 to A3 of our PCR plate, and so on. So if you look at this, you might think that this will need quite a lot of clicking. In fact, this is not true, because our software involves a smart and patented pattern recognition algorithm. And it works like this. As the first step, you click on your source tube, and then you click on your 3 destination wells. You then click on your second source tube, and the software will automatically suggest the wells B1 to B3 as your destination wells. So it will recognize that you want to do triplicates. To fill up your plate, you can simply click on Command, and as the software has recognized the pattern you are using, it will fill up the full plate with only one click. All right, let me move on to a second example. Again, something which you do in the lab all the time. So how about dispensing reagents from, let's say, a conical tube to a plate? A process which you would do in multi-dispense mode, for example, with an electronic pipette. So in a more simplified view, this means taking out liquid from one vessel and putting it into several wells or several other vessels. For this process, we have a command called Reagent Transfer, which allows the distribution of reagents from one well or one column to many destinations. Again, to use this command, after populating the work table, you simply drag and drop the command from the menu on the left to the procedure list. You then choose a tool that you want to use, the transfer type. In this case, we're using the multi-dispense mode, the tips, and the volume you want to transfer. You select your source and your destination vessel, and you choose your pipetting pattern. In this case, our source vessel is our reagent rack. This reagent rack is a modular piece of labware which allows the use of vessels of different shape and size in one position. So here we have, for example, 15 milliliter conicals, 50 milliliter conicals, and 1.5 milliliter reaction tubes on one deck position. So, and for the reagent transfer here, we transfer liquid from the position 1A of our reagent rack to our PCR plate, to all of the wells of our PCR plate. So let me very quickly explain you the difference between pipetting and multi-dispense mode. So if we talk about pipetting, we're referring to a process where you aspirate a given amount of liquid, and then you dispense this total amount of liquid into one destination. This mode allows to use a blowout, which makes it possible to handle also difficult liquids. For example, liquids which contain a high level of glycerol. It's a very accurate liquid transfer mode. The multi-dispense mode works a little different. You take up a large amount of liquid, and this liquid is then subsequently dispensed into several destination wells. This works faster than the pipetting mode and allows multiple deliveries with only one tip, which saves consumables. And as you can see here in this presentation, in our pipetting and also multi-dispense mode, there is always a little air gap at the tip of the tip in this case, and this makes a contamination-free movement of liquids possible, and it's a safety measure to protect your precious reagents and your important samples. One last reformatting example. How about the removal of liquids from a plate to a waste vessel? This is something which you could imagine, for example, if you wash ELISA plates, and a process that you would manually do in most of the cases with an 8-channel pipette to speed up the process. So if we go to a more simplified view, this means that we take out liquid from several wells and we transfer it into one destination well. So the command we use is called pool to one destination, and it allows the consolidation of samples to one well or one column. In the software, again, after populating the work table, you simply drag and drop the command to the procedure list. You select your tool you want to use. In this case, we are using a multi-channel tool, the volume you want to aspirate, and your source and destination. You then choose your desired pipetting pattern, and as you can see here, we are aspirating liquid from the whole plate and transferring it into a waste reservoir. So, of course, you can not only use those different commands isolated, but you can simply combine commands as you would do it with manual steps. So, as for example, you first want to distribute your reagent. You do this with the reagent transfer. And you then add your samples to your reagent. And this you do with a sample transfer. In our epBlue software, this is realized by simply dragging and dropping those commands in our procedure list. You can add as many commands and steps as you want, and those commands will then be performed as been put in the list subsequently by the device. All right, let us move to the next building block, which is a dilution series, something which you do very often in a lab. So this means that you work with stock samples or any stock solution, and you dilute them mostly in a well format with, let's say, a multi-channel pipette to have several dilutions of your stock solution. For this, we have our dilute command, which allows serial diluting of 1 or 8 samples with a common factor, something very useful for standard curve preparation or also the preparation of working solutions. In the software, after populating your work table, selecting the dilute command and the desired volume, here you simply choose your stock solution. In this case, we're using our reagent rack in a different setup with the 8 strip here in the first position. We have then 1.5 milliliter, 50 milliliter conicals, and 3 reservoirs. And we then transfer this to our destination plate, where we do a serial dilution over the full plate, meaning over all of the 12 columns of the plate. Let's now move on to the, let's say, a little bit more exotic building blocks. So first we take a look at so-called cherry or hit picking. This means picking several samples from one plate, which might be in a rather chaotic order, and consolidating them on one hit plate. This is a very tedious and error-prone work if you do this manually. So if we want to take a look how this is realized by the epBlue software, we can simply do this by using our pattern. So here we have to select our source well, so let's say A6, and we move the sample from this well to our destination well, which is A1. And we do this for all of the other samples we want to pick. The problem with this process is that you still have to look in your list and figure out which sample goes into which well and which well you have to pick. So still there is a little option for making mistakes here. So there is a more convenient way to do hit picking, and One thing I want to highlight here is what you have noticed is that our software is also able to work with irregular patterns. So as you see that the pattern from the source plate is chaotic, so the system doesn't want to find a pattern here if you tick this box. Okay, but let's go back to the more convenient way to do hit picking. In order to do this, we use so-called CSV files, which are nothing else than an Excel list, a data list, or a separate value file. So a file you prepare beforehand where you sum up all of your pipetting steps. So how does such a list look? It's a very simple list. So the first two columns describe your source, and the third and the fourth column, they describe your destination. You then choose the volume you want to transfer and the tool you want to use. In this case, the single channel tool for 50 microliter. And you can always give a name for your samples. So once you've prepared this table, you can go to your epBlue software. You populate your work table. Here we are seeing a work table of a different epMotion type, epMotion 5073, which has slightly more deck positions, as you have seen before. And what you do is you program a sample transfer step, and you choose the volume to be 0 microliter. You then choose your source and your destination. In this case, we are pipetting from a 380 well plate to a 96 well plate. You then simply click on the CSV import button. You choose your hit picking list, so your desired CSV file. You choose your CSV import options. In this case, we go for the transfer type pipette, which allows to transfer each sample with the individual pipetting step to avoid contamination. And once you've done this, you can see here in your procedure list that your CSV file is converted into a number of sample transfer steps. So this allows you to very conveniently do hit picking. All right, along the same lines, we will look at the next building block, which is normalization. Normalization describes the process where you dilute samples with different concentration to one defined concentration for all analysis samples. This is one task in the lab which I personally hated, because it involves tons of pipetting steps and it's extremely error prone. So I would highly recommend you to use, if you have the option, automated liquid handling for this task. And for the epBlue software, again, we use CSV files, which is a very fast and convenient way to do your everyday normalization tasks in the lab. In this case, our table looks just a tiny bit different. Again, we have our source and our destination. We have the volume, and we have the tool we want to use for this step. The difference is that in these files, we can give a source for both the samples and the diluent, as you can see for the sample here and for the diluent here. So how you import this into our epBlue software. So again, first you populate your work table. Here we're seeing another epMotion version, which is the 5075 and has the largest deck capacity. So you program a step again with 0 microliter pipetting, and then you choose your source for both the diluent and the samples. In this case, the diluent comes from a reservoir, and the samples come out of the deep well plate. You also choose your destination, and we will consolidate our samples on a PCR plate here. You then simply click on the CSV import button, choose your CSV file you want to import, and then choose your CSV import options. In this case, we are going for the multi-dispense mode, which allows to use the multi-dispense mode for replicates of equal volume. This saves both time and consumables. Once you've done this, you can see that the CSV file is again translated into a number of sample transfer commands, which now use the multi-dispense mode. So all of those commands I have presented you in the last minutes are dealing with the transfer of liquids. But you know that lab work is more than just transferring liquids. So we will now look at the so-called auxiliary commands, which involve mixing, incubation, and transport steps. So the commands we are talking about here is, for example, the wait command, which has been used for an incubation step and allows to wait between two steps you've given in your procedure list. Another command is the pipette mixing, where you can mix a liquid with a pipette just as you would do, for example, with a master mix or with a bead solution. You can here simply choose the number of mixing cycles, the mixing speed, and the volume you want to mix. Another command, very important command, is the transport command, and this allows the automated relocation or stacking of labware with a gripper, which you can see here. So in the software, you simply choose the labware you want to move and to which position it should be moved. We have a pierce command. The pierce command allows to work with sealed plates, either with film or foil. This film or foil is then pierced with a single channel tool to allow the aspiration of the liquids underneath. If you have a magnetic separation device, you can turn on and off the magnet using the magnetic separation command, and you can do this for your choice of time. If your system comes with thermal modules, you can set the temperature for those modules to allow heating or cooling on deck. If your device has a thermomixer, which is a combination of shaker and thermal plate, you can choose your mixing speed, your mixing time, and the temperature. Last but not least, if you have a device with a vacuum chamber, you can choose the pressure of your vacuum and the duration of the vacuum. This is very useful if you deal with filter plates, for example, for nucleic acid binding, washing, and dilution. So now, after we've been through all of those building blocks, it's very important to mention that all of these blocks can be combined in any order and by any number, and this allows to display even the most complex workflows. So if we look, for example, at a PCR reaction mix, you have your template DNA, which comes from either hit picking. It might be normalized, maybe it's diluted, and definitely it will be some sort of reformatting process. You then have your master mix, which might also be diluted and reformatted. And you have your primers, which can be diluted again, and they will be reformatted. And if you bring all of those components together, you have your PCR application. And on the epMotion, you can simply realize this by aligning different commands. Let's take a look at another complex workflow, which is the bead cleanup, which can, for example, be part of the NGS library preparation. And as you can see here in this slide, so you can break down complex workflow into several single steps. So you start with the mixing of the beads into the sample. You do this with the reagent transfer and pipette mixing. You then incubate this. Just use the wait command. You move on and do the magnetic separation. You just transport your plate to the magnet there. You then use the pool to one destination command to remove the supernatant. The next three commands are first the adding of the wash buffer with the reagent transfer. Then you move it to the magnet. You incubate and separate, and then you remove the supernatant. So this washing process can be repeated several times. After you've done with the washing, you usually have a drying step. You do this by waiting and incorporating a thermal module with a temperature step. You then transport your plate back to the magnet, use the reagent transfer and a mixing command to resuspend your beads in elution buffer, and you then do the final incubation and separation by moving your sample and applying a temperature. In the end, you will transfer your supernatants with your nucleic acids to a fresh plate. If we then take also the bead cleanup and the PCR as a building blocks consisting of smaller building blocks, you can think about the most complex workflows. So here, for example, you have a workflow which incorporates a bead cleanup, then there might be several steps in between, then there can be a PCR, maybe some other steps, and in the end you will have another bead cleanup. But as I could show you in the previous slides, you can all break down those steps into simple commands which you can align in the software in your procedure list. So I hope in the last slides I could convince you that programming in epBlue is fast and easy by just dragging and dropping commands. So let me just quickly wrap up the epBlue software benefits. So you have a standalone editor which allows programming at your desktop PC, so you don't have to sit next to the machine all the time. Our graphical user interface is very intuitive and comes with drag and drop programming for both labware and commands. We are using a lot of smart commands which can be understood very quickly. We have a comprehensive labware repository with tons of labware files from different vendors which can be used on the machine. A very important feature is also the 3D method simulation, which you can see down here. So this allows the in silico run of your method to avoid the loss of precious reagents or consumables. And we offer a lot of pre-optimized methods, for example, for nucleic acid purification, next generation sequencing library preparation, and a lot of other applications. But besides the easy programming, automation also offers you a lot of other benefits, and I guess that Carsten will introduce them to you now.
Yeah. Okay, thank you, Tim. I hope everyone could really understand that it's now really fast and easy to program various routine applications on automated liquid handling systems like our epMotion. Of course, there are many good advantages of the automated liquid handling. So they are listed here. The first one would be the versatility of the system. As you have seen from Tim's presentation, with all the different commands, you can build up the building blocks, and with that you can really implement a lot of applications on one of the same systems. So you can do nucleic acid purification, PCR setup. You can do cherry picking, which is also very error-prone task. You can do ELISA preparation, cell culture work even, and one of the most complex but also very tedious and labor-intensive application nowadays is the next generation sequencing library preparation, which is definitely something which is very useful to do this in an automated way. So another advantage is the throughput capabilities of the automated systems. You have seen you can do the reformatting just from one tube or few tubes, but certainly it will help you to increase the throughput and the efficiency in your lab, because you easily can now start to work with plates or with several plates in one batch of your application runs. So definitely the throughput is increased with automated liquid handling. However, you not always have to apply it for really high throughput applications. It's worth doing it as you have seen it in some examples from Tim. Doing the normalization, it's worth doing it in an automated way already with a few samples, let's say 8 or about 24 samples. It's already very reasonable to do this in an automated way already. So you can be a very experienced manual liquid handling device user. However, usually the accuracy and the precision of an automated system is much better if you use an automated system. And here below, we have collected some experiments. They are documented in an application note where the accuracy and precision of the PCR setup by manual or by automated means were compared, and definitely the accuracy and precision of the automated method is much superior. So this is also an advantage of the automated liquid handling, like for instance the next one, which is the error-free or the reduction of human errors during all the pipetting steps. So in a recent study we have seen that there are many reasons why you can have failures during your pipetting. However, in about 70% of the cases here in the data from the recent survey, it is showing that it is due to the pipetting by the user, which is the main portion of the errors in this case. So this is definitely reduced by switching to automation. Another advantage is all the sample tracking and documentation that you need to do in the laboratory. You can imagine that, or you have to document everything in your lab journal, but the automated system is doing this in a very comprehensive and detailed log file, which is provided for each run with the system. So, for instance, there's a tracking for the barcodes or the sample name, or the operator obviously need to log in the system, and with that it is documented. Of course, the time and also the procedure or method which is used, the position of the sample in a plate, from where to what destination do you do the pipetting, and of course the device status is also documented, and all the volumes of the pipetting action are also documented in one very detailed and combined log file. So this is very convenient for you as a user, and definitely an advantage for automated liquid handling systems. The epMotion has really a nice combination of multi-level contamination prevention. So this is done first of all by a complete enclosure of the system, and also with the on-top clean cap where an HEPA filter and an UV light is included. You get further protection of your samples inside of the epMotion. But there are other contamination prevention measures. It is the contact-free or free-jet pipetting mode, which is used with epMotion, or using sterile filter tips, autoclavable pipetting tools that is really a standard for our pipetting system, that you really can take out the tools and send it for not send it. You can send it for calibration, but you can do the autoclavation by yourself. Then we have all our tip qualities. We have various tip qualities, but the PCR clean is one of the highest qualities for our tips. We have a command which is a rinse feature, so this can be used to rinse and clean a pipetting tip after the action before you put it to waste, which can also help to prevent contaminations. And there are other optimizations within the epBlue software. So with all these multi-level contamination preventions, you are protecting your samples and your reagents, which is very important. However, it's also important that you protect yourself during all these routine assays, and also protect any operator from all the biohazardous substance that you might work with. For instance, if you work with blood or other body liquids, and of course we are working with viruses or other pathogens in your laboratory work. So protecting personal protection is also a very good advantage for using automation in your lab. So, last but not least, there is, of course, combined with any automation in the lab, you increase the standardization because you are working with fixed methods which you use again and again, and which gives you a standardization to your work. And with that, you also get an increase of the reproducibility of your results, which is very good, because if you imagine you work with different operators, each operator will have its own reproducibility, but finally the results will differ from each other, and really a good reproducibility means that from several usages you are getting really close in the results. And this is achieved with using automation, increasing the reproducibility of your results. So free up your time with ease of use by switching to automated liquid handling. So it increases the productivity of your work. You will have less errors or failures in the experiments. You need less repetitions of your experiments once they are failed. And with that, you get a better efficiency. And I think it's very clear that everyone has a lot of routine liquid handling tasks every day in his lab, like the reformatting that Tim talked about, dilutions, hit picking, or even the very tedious and labor-intensive normalization steps. So this is done in pretty much every laboratory, day in, day out, and these are really nice and simple applications that are easy to transfer to automated liquid handling. But it can also be more complex applications, like the NGS library preparation, that are really very reasonable to automate. Because, for instance, it's manual work will take about 2 days of hard pipetting and very concentrated work. It's very tedious and it's very repetitive. And with automation, you can really reduce this to only 1.5 hours of hands-on time with the help of the epMotion. And this really frees you up to spend more time on other sophisticated scientific work in your lab. Okay, so with that, I would like to come to a short introduction to our product portfolio. So we have three different types of epMotion systems. So from the largest one, which has 4 deck positions, but you can have virtual deck positions which you use in the programming, and this can be indefinite. So you can program methods, and in between the software will ask you to replace labware in the system before the run continues. So with that, you really can increase the capacity and the complexity of the applications, even with a small system. But we also have the next level of instrumentation, which has 6 positions, and also the capability of using a gripper device. And then we have a set of larger systems which have up to 15 deck positions, and then also waste position is available on all the devices, and up to 4 positions for exchanging the pipetting tools, and of course the larger systems can also handle a gripper. So with the different capabilities, it is also different usage of applications. Usually the small systems, the epMotion 5070, is used for PCR setup and general liquid handling task, while the 6-position system, the epMotion 5073, is used for a bit more complex methods like nucleic acid preparations, bead-based purifications, or also a lot for PCR setup. And then the larger system, that is really useful for the more complex and for applications with a little bit higher throughput, like the ELISA, cell-based assays, and definitely the NGS library preparation. There you need a lot of deck space to work with all the different kit methods. So we have a lot of accessories available for our systems, and over the 15 years of epMotion, we really added a lot of things to it, and we have about 8 different pipetting tools, single or multi-channel, and with that we can cover a broad range of volume. We can go from 0.2 microliters up to 1 milliliter. So this is really a broad range which you can use for various applications. We have a gripper device to handle labware in the system, and a lot of different racks, thermoadapters, and various others accessories that can manage to implement all the various applications on the epMotion system. And of course, we also have a good set of consumables, so tips and reservoirs to use. In addition, we also offer all the needed support services like maintenance, IQ, OQ services for our instrumentation. Okay, that should it be with a short introduction to our product portfolio. With that, I would like to summarize today's webinar. So I hope you could see that there are many applications and routine tasks that are reasonable to automate. And all of these applications consist of workflows, and that can be defined in certain building blocks, easy to transfer to the epMotion. And I hope that Tim could show you that with the way the programming is done, that is very intuitive to do, and that a setup of certain applications is also really quick to do on the epMotion system. So in addition to the easy and intuitive programming of the system, I hope that I also could show you some other good advantages of automation, which makes it reasonable to switch to an automated system device. Okay, with that, I would thank you for your attention, and Tim and me are now available for any questions that you might have.
We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the green Q&A button at the lower left of the presentation window. Type your question into the box that appears on your screen and click the send button. We'll answer as many of your questions as we have time for. Let's get started. We have an ample number of questions already coming in from our audience members. Gentlemen, you demonstrated that the reservoir rack can hold multiple types of tubes, vials, and other things. Can the tube racks hold different ml tube sizes?
Yeah, a very good question though. Yes, our racks can hold tubes of different sizes. So 0.5 ml tubes, 1.5 ml tubes, and 2 ml tubes, and it is also possible to have a mix of different tube sizes within one rack for one application.
We call this mixed rack labware, and in the labware selection, which you can do in the software, you can find a selection of predefined mixed racks, which you can use for your application, providing 0.5, 1.5, and 2 milliliter Eppendorf vials in a certain combination.
Thank you. And can the pipettes be calibrated? And if so, does Eppendorf offer this service?
Yes, of course. Any pipetting device needs calibration after a certain time. The tools have an electronic chip in it, so they will recognize any pipetting action, and they will notify the user after about 200,000 pipetting actions. There will a message pop up and ask for calibration. So, and then it depends on the country, but in all countries we definitely provide calibration service, and in most of the countries we even provide on-site calibration service. In some smaller countries, you have to send in the pipetting tools for calibration in one of our offices. Yeah, but definitely calibration of pipetting devices is very important.
Thank you. And what is the lowest volume you can pipette?
The lowest volume we can pipette is 0.2 microliters. So this effectively allows also to downscale certain applications, for example, PCR or qPCR setups. Just as a reminder, the highest volume we can pipette within one transfer step is 1 milliliter. And we cover this range from 0.2 to 1,000 microliter with 4 different tool sizes.
Great, thank you. We have an amazing audience today and a comprehensive set of questions coming in. Here's one. How do users know which tips to choose?
So this is done one part by the software, where the user has to define if they want to use a filter tip or a non-filter tip. So that is done in the setup of the properties of the sample transfer or reagent transfer, and then it depends on the selected volume and the dispensing tool, and the instrument by the software will then pick the right tip from the tip boxes provided on the deck. So that is not something that need to be programmed by the user. The system and the software will do this absolutely automatically, and it will take the next available tip. So if it's an 8-channel tool, it will take the next row. If it's a single-channel tool, it will remember where's the next tip available in the box, and by that it will use up the complete box. tips, and of course can go to the next box of available tips if required. So that's done by means of software. The only thing is to define if a filter tip is to be used for this pipetting action or a non-filter tip.
Thank you, gentlemen. And our next question is, what does this look like if you are transferring 9 samples?
I assume that you are referring to a certain slide, which I will quickly open. So that one over here. You can easily transfer also 9 samples. So this would then mean that the sample from this source vessel goes into A4, 5, and 6. And the system would also recognize this automatically. If you tell it before that you want to use 24 samples, for example, in total, and you're also not bound to do your triplicates in this horizontal orientation, you could also simply do it vertically. So it's your choice, and believe me, the pattern recognition works really well. So the pattern you are choosing will very, very, very likely be recognized by the system.
Thank you. Our next audience question comes in 2 parts. The first part is it possible to perform different dilutions using different factors? And then can you dilute vertically, not only horizontally across the whole plate?
Yeah, very good question. Also something which you see in a number of applications. Yes, you can dilute using different factors, but not within one dilute command. So that would mean that if you want to dilute with different factors, you have to use several dilute commands depending on the diluting factor. But for one dilute command, you can go for every factor that you want, basically. For the second part of the question, you can also dilute vertically. You can also dilute horizontally over a plate. Yeah. Vertically and horizontally, I'm sorry. Yeah. So both directions is fine. However you want to dilute, it will work.
Great, thank you. We have time for one more question from our audience members. How do you help a customer know when it is appropriate for them to switch from pipettes to epMotion?
So that depends on the application the customer is doing, and usually it is worth speaking with our sales representatives about the application and the pipetting that is done, the routine task in the lab. As we demonstrated, there are a lot of routine tasks where pipetting is involved, which can definitely be transferred to an automated system. And we are happy to talk with the customer if the application is feasible and what would be the required instrumentation to use it for. And then in a detailed discussion about the application, our salespeople, our specialists will help to set up the right configuration. And with that, we can even then have a demonstration of the software, or even place a demo system inside of the lab of the customer if really required. But we have many routine applications where we can have good argumentation and convince the customer to provide the budget for one of our epMotion systems. So definitely the next step is to talk with our product specialist in sales.
Thank you so much. And gentlemen, thank you for your presentation today. And we know that you will be answering all further questions via email. Do you have any closing remarks you'd like to provide the audience before we close today?
Yeah, thank you for the attendance. It was amazing to see that so many people were interested in today's webinar, and we hope you enjoyed the performance of our talk. And also thank you, Susie and Christy, for the good moderation of this event. And with that, thank you, everybody. And yeah, let's close the session.
Thank you so much, gentlemen. And I'd also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through April 2018. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thanks for joining us. We hope to see you again soon. Goodbye.
Goodbye.